students, today in this video I am going to explain you about quick revision of Zoology Intermediate Practicals. Follow the instructions, you can score maximum marks in your practical exams. Once you receive hall ticket, students, please look the time slot of your practicals date and time, whether it is afternoon or forenoon, right? This is very important information I want to give, so please follow this. So zoology practicals will be for 3 hours, students. Maximum marks will be 30. It is having 4 important aspects. First part is to observe the displayed system in the form of unlabeled model or chart. You have to identify the system. What system is that? Draw the neat and labeled diagram. And students, don't get panic. You have to write minimum 4 parts. No need to write each and every labeling, but write correctly, students. I would like to show you all the charts now. Total 11 charts are there in your zoology manual. Any one may come in your exam, and they are. It can be cockroach mouth parts. This is the model of the chart, students. It will be displayed in your examination hall. Your work is, you have to identify that this is cockroach mouth parts. And minimum you have to write 4 labelings. You can see here I have labeled many. At least you write 4 to 5 labelings. Like submentum, labrum, maxillary pulp, mandibles, hypopharynx, cardo, stipes. In general, this is maxilla. And this is lower lip or labium. So already you have studied in your junior intermediate, and this is one of the important 4 marks questions, students. Observe this, cockroach mouth parts. Another chart, human digestive system. This chart will be displayed in your examination hall if this comes in that exam. And it is very easy, students, human digestive system. You have to draw a neat diagram. Use color pencils. Avoid sketches, but use color pencils. And labelings, basic simple labeling, students. Esophagus, stomach, pancreas, small intestine, gallbladder, liver. You can very easily do this. Don't make a mistake in labeling, students. Next, human venous system. Students, difference between human arterial and venous system. First, you identify the color. If color is blue, it is venous system. If color of these blood vessels is red, then it is arterial system. Now, see, the chart is displayed. You have to draw this diagram and start labeling. Basic labelings, external jugular vein, superior vena cava, heart, you can identify easily, lungs, inferior vena cava, hepatic portal vein, common iliac vein. Students, be careful while labeling and be cautious with spellings, because this is the first question. You are drawing this on sheet 1. Students, be careful. Please maintain neatness. Draw neatly. It is very easy. Draw colors and at least write 4 to 5 labelings. If you write more labelings, it is well and good, but write correctly. Next, you can see this earthworm nervous system. Very easily you can identify. Already you studied for board exam, NEET, EPSET. Draw the diagram, students. So you be cautious, students, while writing the numbers of the segments. Be very careful. You need to be particular, students. Right. So these are nerves, suprapharyngeal ganglion, subpharyngeal ganglion. Of course, this is nerve ring, students. Nerve ring. And segmental ganglion. Yes. 1, 2, 3, 4 and 5th labeling. And simple diagram. So this is, of course, a digestive system, and this is very important what you have to draw. Both things you have to draw, because nerve ring is surrounding pharynx. Suprapharyngeal ganglion, subpharyngeal ganglion. Students, be cautious. Next chart, human urinogenital system, students. Human urinogenital system. See, for second year, because you are second year students, as you know that human male or female reproductive system is important for board exam. So you are already perfect with the diagram. You choose any one diagram, students. Already you are perfect in human male reproductive system and female reproductive system, human. But students, write urinogenital system, which is the heading in manual, board of intermediate manual. We have to follow that heading. Right. So draw the diagram and labelings. Simple labeling, students. Urinary bladder, seminal vesicles, prostate gland, epididymis, testis, urethra, penis, glans penis, urethral urinogenital aperture. Very simple labeling. Everyone, almost students in Telangana or even in Andhra, they are preparing this male or female reproductive system, which is important even for IPE. Right, students? Next chart, female urinogenital system, students. Any one diagram you can choose, but draw neatly. Right, students? Uterus fundus, uterus, and all the 3 layers, perimetrium, myometrium, endometrium, ovary, fimbriae, infundibulum, ampulla, isthmus. These labelings, basic labelings, everyone can write, students. And see, the diagram is already displayed on the chart. And you can use these colors, or you can use your interested colors. But make the diagram attractive, students. Next chart. Cockroach nervous system. Cockroach nervous system. While drawing cockroach nervous system, please be careful with 3 thoracic ganglion. 1, 2, 3 and abdominal ganglion. Students, in 5th abdominal segment there is no ganglion. Examiner will look into that point. Please be cautious. Like 1, 2, 3, 4, 5, 6 abdominal ganglion. A1 present in 1st abdominal segment. A2 2nd abdominal segment. A3 3rd abdominal segment. A4 4th abdominal segment. In 5th abdominal segment ganglion is absent. Again in 6th and in 7th segment. So observe this chart. Draw the diagram neatly, students, and don't forget to label correctly. Like I am revising one time these labelings. Cerebral ganglion, which is supraesophageal ganglion. Below, subesophageal ganglion. And these 3 are thoracic ganglion, and these are abdominal ganglion. Here, the larger one, 6th and last abdominal ganglion. Students, please observe the points what I said. Please do follow. And next chart, earthworm digestive system, students. Again, in this earthworm digestive system, please do practice and come earthworm digestive system, because you have to draw segments carefully, students. How to draw the segments? First, draw lightly with some light color pencil. Later, make it thick and write segments very clearly. Especially gizzard present in 8th segment. Now here, prostomium, buccal chamber, pharynx, see 4th segment, esophagus from 5th to 7th segment, gizzard in 8th segment. And clitellum, don't forget to draw clitellum, which is present in 14th, 15th, 16th segment. It will give an additional attraction, students. Same like that, intestine from 15th segment to last segment, intestinal CK. Even you have to draw this, intestinal CK. Right, students. Next. Cockroach digestive system is another chart. A simple diagram. Please do practice this also. And this chart will be displayed. You have to see and draw. First, you have to identify what is this dissected animal. Yes, this is cockroach digestive system. And labelings: salivary glands, esophagus, crop, gizzard, hepatic CK, mesenteron, Malpighian tubules, rectum, anus, and all. As I said you, 4 to 5 minimum labelings. You can write more labelings, but please be accurate and correct labelings. You have to label, students, or else it will look as an error. Right. Next. As I said you in the beginning, students, human arterial system. Human arterial system. As I said, how to differentiate arterial and venous system. Venous system, blood vessels are blue in color. Arterial system, blood vessels are red in color. Right. See, chart will be displayed. You have to see and you have to draw. First, you have to identify. For identification marks, I'll now say you the scheme of marks award also. Like this is common carotid artery, subclavian artery, aortic arch, dorsal aorta, lungs, heart. And this one, celiac artery. Celiac, C-O-E-L-I-A-C, celiac artery. And the one which is going to kidneys, renal artery. And this one, common iliac artery. Right. So out of this, you can choose any 4 to 5 or even 6 labelings, whichever your comfort, students. Right. Now coming to earthworm spermatheca, in most of the cases, we have observed spermatheca will be experienced by one or other batches. For more batches, we have seen spermatheca will be coming. Again, here also be cautious, students, while drawing these segments. First you draw lightly and later make it dark, thick, and segments 6, 7, 8, 9. These segments are very important. While drawing this, be cautious, students. Spermatheca should be drawn in this way. Right. See, these both diverticula are facing to each other. 4 pairs attached to 5 by 6, 6 by 7, 7 by 8, 8 by 9 intersegmental septa. And you have to compulsory draw this glove shaped structure. So one is diverticulum, other one is ampulla. Right. So students, that's about the charts, which is very important part. The moment once you enter into your examination hall, first you have to do this task. See, scheme of evaluation, 6 marks will be maximum for this question 1. In this, identification carries 1 mark. Like whether it is mouth part of cockroach or spermathecae or digestive system, it carries 1 mark, whatever it may be. And drawing neat diagram, 3 marks. And labeling, 2 marks. Scheme of evaluation. Evaluation is very important. You please note down this point also. And accordingly, students, you can perform in the exam. So the moment you completed your first question, which is drawing diagram and labeling. Which is first one, drawing diagram and labeling. You have to turn the page, that additional sheet which will be given by the examiner, and go for second important part. That is, the question will be written already on the board. Identify the presence of sugar. For example, in the given samples A, B, C, D. You have to write principle, procedure and result. This carries maximum 5 marks. Writing principle, for writing principle, 1 mark. Compulsory everyone should write principle, and procedure 3 marks, and after drawing that table and finally when you write result, it carries 1 mark. I would like to display that also to you, students. Yes, I took it. This is demonstration of digestion of starch by salivary amylase. Principle. See, compulsory you have to write this principle, students. Enzyme salivary amylase present in saliva break down the starch. It hydrolyzes into maltose and dextrins. Enzymatic activity of salivary amylase can be tested by detecting the presence or absence of starch. And next, apparatus or requirements, procedure and result. So another physiology experiment is starch, identification of presence of starch in given sample. Very easy. You have to take one sample in all the 4 textbooks or 4 test tubes. Please take sample in all the 4 test tubes and add iodine to that. You can see the color change. If it turns into blue in color, then the sample in which you took the test tube, for example in A, it turned into blue color, then starch is present in sample A. So like that, the same thing, you have to draw the table and you have to write the result. Compulsory you have to write the result. Every point, aim, principle, requirements, procedure, result, table, every point is very important. Same like that, identification of presence of glucose in the given sample. Identification of presence of lipids, that is either fat or oils in the given sample. In all these things, please go through principle properly. And another last one, that is identification of presence of albumin in the given sample. Students, I am showing you one model experiment. Identification of presence of albumin in the given sample. For that, you have to take carefully 4 to 5 ml of nitric acid and add 1 or 2 drops of the sample in that. If you observe the formation of white ring, this indicates the presence of albumin in the given sample. So this is very easy experiment, students. Hope you understood. Thank you. Students, question number 3, part 3. Identify the spotters which are displayed in your lab, in your examination hall. A, B, C, D, E, F, G. Right. So what you have to do? You have to first identify and later you have to draw a rough or a good diagram and label it. Minimum 2 labelings are needed, students. So in this, for your easy understanding, I would like to display quickly all the slides and specimens. In A, invertebrate slide will be there. In B, invertebrate specimen from Euspongia to leech will be there. In C, invertebrate specimen from Scolopendra to Asterius. In D, histological slide will be there. In E, mammalian slide will be there. In F, vertebrate specimen will be there. And in G, joints will be there. I would like to give a quick revision and make you understand how to write the points, students. How to write the points. Right. First, let us focus on this. Euspongia, bath sponge, a rough diagram. If possible, write phylum name, phylum Porifera, and write 5 easy points, students. Like for example, it is called a common bath sponge. It has many pores called ostia. These ostia opens in the body cavity called spongocele. Canal system is present inside and it contains spongin fibers. By observing above characters, it is identified as Euspongia, commonly called as bath sponge. Right, students? See, what is the scheme of evaluation for this? For identifying, you will get a half mark, students. You identified that this is a Euspongia, bath sponge. Then you got half mark. You draw a labeled diagram, you get half mark. If possible, if time permits, use colors to the diagram. Write minimum 2 points. If possible, write 2 to 3 correct labelings. And identification points, 1 mark. You have to write 4 to 5 identification points. This will attract the examiner. They will award you the complete 2 marks. Total 7 spotting will be there. 7 into 2, 14 marks. As I said, students, I would like to show you these spotters and specimens. Right, here next one. Aurelia or jellyfish belongs to phylum Nidaria. You can use this diagram. Metridium or Adamsia belongs to phylum Nidaria. Already you studied in your first year. Liver fluke belongs to phylum Platyhelminthes. Taenia solium, tapeworm, again belongs to phylum Platyhelminthes. Ascaris male and female, be cautious here. If the posterior part is curved and it is shorter, then it is male roundworm, belongs to phylum Nematoda. If it has a long tail-like structure and posterior part is not curved, then it is Ascaris female roundworm. Half mark for identification, half mark for drawing a diagram and 2 labelings, mouth, excretory pore, gonopore, anus, and writing 4 to 5 identification points. Don't forget at the end to write that by observing above features, it is identified as so and so. It is identified as Ascaris male, Ascaris female. Nereis sandworm, phylum Annelida. Earthworm, phylum Annelida. Leech, phylum Annelida. Now, see, in C, invertebrate specimen. See, Scolopendra, phylum Arthropoda. Jewelus, phylum Arthropoda. Paleomon, prawn, phylum Arthropoda. And a simple diagram, if you are facing difficulty in drawing prawn, students, a simple diagram. So a simple like this, you can draw a rough diagram, but you have to label like compound eyes, antennae, antennae, biramus appendages, carapace and all. Arania spider, phylum Arthropoda. 4 pairs of legs are present. Very important point, students. Pelamnius Arthropoda, antennae are absent. Pedipalps are present, student. Pedipalps, very important point. Again, 4 pairs of walking legs are present. Silk moth, Bombyx mori, Arthropoda. You can write commercial importance of this silkworm. Honeybee, again phylum Arthropoda. You can write commercial importance of this honeybee. It is a colonial insect. It secretes honey and all. And 3 categories of moths are present. Worker bees, these are sterile females, queen bee, fertile female, larger in size, drones, males and all. Pila, apple snail, phylum Mollusca. Simple diagram. Like this. Be confident, students, while drawing diagram. Unio, water mussel, phylum Mollusca. Asterius, starfish, belongs to phylum Echinodermata. Again, part B, histological slides will be there, students. In that, either squamous epithelium, columnar epithelium, transverse section of cartilage, TS of bone. You can expect, like in squamous epithelium, you can draw the cells like this, large nucleus, and the cells are laid on basement membrane. In columnar epithelium, the cells are long, slender, nucleus is present, basal. And where is it located? A blood smear of mammal. And what are the cells present? You observed in the blood smear, like RBC, WBC, in WBC granulocytes, like eosinophils, basophils, neutrophils, agranulocytes, like monocytes, lymphocytes, everything. And striated muscle, multinucleated, smooth. This striated muscle or skeletal muscle, that point you have to write, students. Even smooth muscle, unstriated and uninucleated. These are involuntary muscles. And section E, mammalian slides. In the TS of stomach, TS of intestine. In TS of intestine, you have to draw this villi and microvilli. In TS of stomach, you have to draw gastric rugae. And LS of kidney. This is 4 marks important diagram for second year students, intermediate students in zoology. Transfer section of liver. Functions of liver is again very important diagram. Next. TS of pancreas. TS of testis. TS of testis, students, again it is important for 8 marks question in board exam. And TS of ovary. Section F will be vertebrate specimens. Like pigeon belongs to phylum Chordata, class Aves. Body covered by feathers, streamlined body. Eyes are provided with mobile eyelids. 4 limbs are modified into wings. And bipedal animals. Digits end with the class. Cloacal aperture is situated in the posterior part of the trunk. Easy 4 to 5 observation points you have to write. Rabbit, Arctolagus cuniculus, phylum Chordata, class Mammalia. Right. And coming to vertebrate specimens, Scoliodon, and you have to draw the diagram, students. I'll show you a simple diagram how to draw. Rough diagram, see. Right. Scoliodon. Same like that, Labiorohita, Rohu. Scoliodon is cartilaginous fish, Labio is bony fish. Katla, Bocce, and these simple points I have provided you in my video. Sirhinus mrigala, Yarramosu or mrigal. Frog, Rana tigrina. Anyhow, you have studied about frog features in your first year. Sea snake, Hydrophis. I have provided you very simple 3 to 4 points, and at the end you have to write by observing above features it is identified as Hydrophis. Naza. While writing this Naza, this species name should be written in small letter. Right. Here neck is expandable, head is covered by shields, hood contains binocellate mark. It is poisonous and poison is neurotoxic. Russell's viper or Vipera russelli. Coming to mammalian joints, ball and socket joint, Observe the diagram, hinge joint, pivot joint and gliding joint. So simple 3 to 4 points, and you can add one more point. By observing the above features, it is identified as the zones of ball and socket joint or hinge joint or pivot joint or gliding joint should be written. So coming back to the slide, students. First slide. So once you complete this, your practicals are completed, and for your record, 5 marks will be awarded by examiner. So by following all these tips and techniques, what I have said you about the scheme of evaluation and the questions, you can score maximum 30 marks. All the best for your practical exams, students. Thank you very much.
MS Education Academy, preparing world-worthy individuals.